me share a link. In the chat, and then hopefully with kind of your comments and the input of others, I can make changes over the next week, and then it's ready to submit to PCO, Precision Oncology. They did I did send them an abstract and a cover letter for kind of a pre-submission evaluation, and they gave me the green light to apply, or not to apply, to submit. So yeah, the figure count is within their limits, and then the word count is within their limits. So yeah, there's room to add stuff if there's any gaps that you see. Kind of some bigger changes that I made since last time you saw this paper was I wrote up a little bit more of a formal method for cutoff selection, and then I found so I changed the cutoffs a little bit, and I found immune gene sets that gave me some really nice enrichment and depletion plots that I think told a good story. And you'll see them kind of at the bottom.
This one?
Yeah, so these signatures were specifically derived from lung squamous and lung adeno. What I was using before were kind of like general immune signatures, but these were specific to these tissues. So I thought they were more appropriate, and they also included PD-L1 and CTLA-4, which I thought kind of added some more oomph.
So where did you find the tissue specific?
I can link the paper. It was a paper from, I think, 2022.
Maybe you can pull up that paper and
Yeah,
share your screen. I
yeah,
want to look at that to see if, where it came from.
yeah. Let me find it. 1 second. Sorry, just give me 1 second. Sorry, 2017, not 2022. Let me pull up the full text. All right. So, I mean, some of it was so basically these signatures were studied in adeno and squamous, and had a little bit more tissue specific literature evidence, so to speak, compared to what I was using before. And it ultimately gave me a better result, so I stuck with it. So this is what it's called. It's published in 2017 by this group. They used some public signatures, and let's see, what else did they do?
Oh, so it's a public signature, but they used in lung TCGA. But it wasn't generated from.
No, sorry, I misspoke.
Yeah.
Let's see if they have any figures that illustrate.
Okay.
Go ahead.
This is their signature, cell signature. Okay.
They also reference this other paper that I think that they adapted the signatures from, Binte et al. And so they looked at, the immune cell signatures. They looked at interferon, And then they looked at MHC expression, and then they looked at individual genes, PDL1, PDL2, PD1, and CTLA4.
Oh, so they mixed it all together.
Yeah.
And okay, so
Which I thought was kind of stronger than what I had before, because
so you used the exact same landscape signature here?
yep, the same
Okay.
gene sets.
Okay. Okay, good, good. You can unshare.
And then you'll see the kind of the enrichment result that came out of it. I thought it was a lot stronger because there was an enrichment for CD8 T cells, and then in squamous there was an enrichment in CTLA and PD1 or PDL1 in if you scroll up just a little bit in the low ITH group, which in this case had shorter progression-free survival. So that was kind of a nice story to say, hey, look, you know, this group has worse survival and it has enriched CTLA4 and PDL1 and depleted CD8s. But kind of throughout this, there would be kind of like, for example, the 3 findings: enriched CD8s, depleted PDL1, depleted CTLA4, was very pro or rather anti-tumorigenic or pro-immune, while TH2s are actually considered pro-tumor. So there was and I write about this in the discussion, there's kind of these pro-tumor and anti-tumor forces. So it's kind of interesting in that regard.
So just for you know, people who are not familiar with this project, the title is Differential Impact of Intratumoral Heterogeneity, ITH, on Survival Outcomes in Early Stage Squamous versus Adenocarcinoma of Lung. And then we say TMB is important in terms of, you know, low, high TMB, ITH. So according to TMB, ITH effect can be a little bit different. So we classify them into here.
I had to add I had to add this diagram for JCOPO or PCO, JCOPO.
Okay. So squamous, adeno, about 350 each. And then among them, we only got low TMB, right?
Yeah.
We excluded high TMB patients, and then we sorted them by ITH. So everyone's low TMB, but high ITH, low ITH, and adenosquamous. So in this 4 different groups, what are we trying to learn? So the reason why we excluded high TMB is that we know high TMB is associated with higher chance of having neoantigen and higher immune response. So we want to take out that factor and see what is the impact of ITH in low TMB group, and how does that play interaction? How is that interaction different between squamous and adeno, right? So that's the kind of question we're asking. And then in the survival outcome, if you look at squamous first, this is PFS and OS.
And that's
high ITH group does better. In adeno is the opposite. And why is it? We don't know, but we just left it. We don't know. This is just low TMB.
That's just low. So that's the first 4 were ITH across the entire set.
Oh, this is entire set. Okay.
And then
is low TMB. Okay. In low TMB, squamous is same. And it's same, right?
Yeah, it doesn't change.
It doesn't change.
The PFS just increases a little bit in that group.
Uh,
Like
so
the difference between the two groups clinically increases. P value is still significant.
yeah. So we try to explain why is there, difference between adenosquamous, and we talk about driver mutation difference. And then this is low TMB squamous, right?
Correct.
And then ITA, high ITH group seems to have high CD8. Did you do this for adeno or no?
I did in the next figure. I just split the figures because I don't know, maybe I could combine them, but
I think you should combine them because you combine them in the survival, right?
right,
So
yeah.
I just think you have to just be combine it, do same thing, A, B, C, D, whatever you want to do.
Okay, yeah.
Yeah. And then, well, I guess this is 4 figure and this is 3 figure.
Yeah, there just wasn't a fourth one that
I
was
see
different.
what you mean. It's 7 figures.
Yeah, I just forgot. I guess I forgot to label those with A, B, C, D, but
Yeah.
I'll change that.
Yeah. Label it A, B, C, D, but I think it's fine to have 7, but I think it should be combined.
Okay.
And just label it adenosquamous here as well, so the people know. But anyway, in adeno, high ITH means low CD8. And I think in squamous, it just makes sense. It's, remember that the cell paper that showed lot of subclones,
Yeah.
meaning, so a lot of subclones had a negative impact on immune response. Immunogenicity goes down if you have a lot of subclones. That's like the preclinical evidence we have. But in let's say, so that means high ITH. If you have more subclones, then it's less immunogenic. So that's more squamous, adeno story.
Yeah.
Less immunogenic. But in squamous, somehow that's the opposite. That low, wait. No. Squamous is
The high group does better.
right. Squamous is more, I guess, not driver mutation driven.
Yeah.
More, lot of different clones. If you have more subclones, then you are more this is high ITH. So I guess
There's
more
more
clones,
antigens.
yeah, more antigens. In adeno, I think just having driver mutation present and just having subclones may mean something opposite, that it's less immunogenic. But then you have more B cells. That's interesting. But the B
Yeah.
cells and T cells does not go together.
Right.
But anyway, we're not trying to make conclusion. We're trying to kind of show okay, the difference.
Yeah, I added these as a supplemental. These are generated by that R package that does the cutoff selection. The reason I included these because other papers often included plots of hazard ratios and cutoffs or p-values and cutoffs. So I thought that this was kind of in line with what other papers have done. The idea here is that every one of these peaks is a possible cutoff. And so what I ended up doing is I ended up testing these peaks, and then of those, picking the cutoffs that had the lowest p-values, the more balanced sample sizes. And this is just TMB. I kept that out of the main body, main
Mm.
figures.
So
Because
this
it
is
wasn't.
using 10.
Yeah.
This is
So there were the clinical cutoff of 10 and then the cutoff we selected. And what's interesting in squamous cell, the selected cutoff did yield a statistically significant result, but in adeno it did not.
I see. So in squamous, the more neoantigen theory is more relevant, I think.
Yeah.
Antigenicity is important versus adeno, it's not.
Right. One thing that I have data for, I just didn't include it here, is I could also do neoantigens or predicted neoantigens. If we want to kind of discuss this neoantigen theory. I just don't remember what the results are. I do have data though for it. And I think it's something I just downloaded from another paper because it's TCGA, so there are a lot of groups that have done neoantigen prediction for TCGA, so it's easier to find that.
Yeah, I don't think you need it. However, in the discussion, you could say, I mean, you've ran it before, so you could say when we ran neoantigen, we did not see any. You could kind of add that even in the result section when you do like talk about TMB.
Yeah.
We also ran in neoantigen burden. We did not see any statistical significant.
From what I remember, it just correlated to TMB, which is kind of what we expect.
Yeah, so maybe just show that. Yeah.
Okay.
But not necessarily graph. And you're gonna include PyClone file. Yeah.
I thought it would be important. They did want to see it, I'm guessing.
And remind me how you calculated the ITH.
So I ran PyClone on every sample basically 9 times. There's a bit of randomness to the way it clusters, and so
Right.
I did 3 runs a day for 3 days. And then I took that and I counted every clone that had 2 or more mutations in it. And then I averaged that across all the 9 runs. The kind of the one topic I didn't discuss here that I thought maybe a reviewer might want to see is kind of more strict criteria for clone kind of selection or clone inclusion. I know other groups have done that. But that's kind of a more detailed kind of thing that may not be relevant. I don't know.
Okay. Did you change any discussion part?
Yeah, so I rewrote the discussion a little bit for these new cell types that were enriched. There were a couple driver genes that were changed, so I wrote a little bit about that. And included a couple more sources.
Okay. So
I just don't have a conclusion, which I probably should include.
yeah. So TMB has been actively utilized to stratify patient. Tumors with TMB are considered good responders to IO therapy, while their no TMB counterparts are not. We have made use of PyClone to estimate in stratify low TMB patients and evaluate their survival outcome. I think PyClone is too methodological. So you
Okay.
could say we utilized ITH to
Okay.
stratify low TMB patients and evaluate. Yeah, our work shows. You can say here we demonstrate that the differential impact demonstrate the differential impact of ITH on survival outcome. Yeah, in lung squamous cell carcinomas. Furthermore, patients with low TMB and low ITH have an altered immune. They may have an impact on survival. So this is like, we don't even understand how,
Yeah.
so furthermore, so this is just like hypothetical. So just say, in addition, we demonstrate that low TMB and low ITH was associated with differential, immune microenvironment
Okay.
in lung and adenosquamous were different. The impacts were different, right? So as a differential impact, adeno versus squamous. Just say that, and that should be fine. And then methods.
Methods should be right after intro.
So I guess what is this? Okay.
Methods up here. Yeah.
Yeah. Here report for the first. So explanation in contrast driver gene to a microenvironment. So so I think you need two more paragraphs. One is the strength of our article is that number one, it's TCGA, the sample size is pretty good.
Okay.
And this has a database that allows us to look at ITH with copy number, and also gene expression allows us to look at the immune microenvironment. So we were able to look at that in different angles. So
Yeah.
that's sort of the strength that we could stratify by TMB and ITH.
Yeah.
And the limitation is that I guess
I think in a lot of ways it is PyClone because of how inconsistent it can be.
yeah, just say, well, we don't want to kill ourselves.
Right.
We just say there's an inherent limitation in applying assessing ITH in terms of how clones are classified, or just write it
Yeah.
more vaguely.
Because
And
there isn't there isn't kind of an established clinically approved method. So
right there. So there's no gold standard in
yeah.
ITH assessment. And every method has limitations or something like that. So that's the
Yeah.
one limitation. The second is this is all early stage, so it's like we're not able to assess any like effective immunotherapy. Most people
Yeah.
wonder what that is, but so we cannot extrapolate, kind of
There's
generalize
no treatment
our
here. Yeah.
data. There's no treatment selection implication here. and just say this is hypothesis generating data. And we and then I think and so that's the strength and limitation. And then the next summer is like, in summary, we hear, just say for the first time,
Yeah.
just say, just like what the abstract said, because I don't think there any papers show that ITH means something different
Right.
in terms of survival and immune landscape between two different subtypes. And this implies that this might have therapeutic implications, or biomarker implication, or this is hypothesis generating and may imply the role of ITH as a biomarker, and its prognostic role as a biomarker may be different between adeno and squamous.
Yeah.
Something like that, you could sort of end so that conclusion. So a
There
lot of
are,
reviewers just like to read
yeah.
the last few sentences. Yeah.
The other thing I was in here, there are papers out there that show differential impact of other biomarkers, cell types, and other scoring systems that have been used. So do you think it's worth mentioning other literature that doesn't necessarily, kind of, although it's not ITH, it's other biomarkers too.
Yeah, I think because this paper is ITH, I think we can just focus on ITH.
Okay.
It's the first one showing ITH. The potential of ITH being a prognostic biomarker, and that differentiates adeno and squamous. So yeah, with that, I think you're almost done.
Great. And then since that last summary paragraph, to me sounds kind of a conclusion. Do I need a separate conclusion section?
No,
Or
that is conclusion.
that will be the conclusion.
Yeah,
Okay.
so you don't need to write. Well, what I don't know, but JCOPO, does that require
No.
conclusion?
So everything that you see here underlined is what they provide as instructions for original reports. And they don't mention any structure other than having a title
Then
page and an abstract. And
you don't need like conclusion title, like end of the discussion paragraph is fine.
okay.
Okay, so with this, I know we're still in the process of getting raw garden files. So Liam, can you update me on where that is?
Yeah, so we had a meeting with them with Stas last week, I believe, right? And we were able to go over what we're looking for, and then we received approval and everything. They said they're going to be pulling the data for us, and Stas gave them like the exact kind of variables and data sets that we're looking for, with like the raw files and things like that. And they said they'll pull the data and get back to us in a couple of weeks.
The strange thing is, she said that they can't give us, they don't have raw data. They only have like what they report in the reports, which we can try and make that work. But it just seems kind of strange to me, because when we worked with Guardant a couple of years ago, they had no problem generating the kind of PyClone input, and they had raw data. But for some reason, I don't know if we're talking to a different group or their systems have changed. But anyway, sorry to cut you off, Liam. I just wanted to mention that.
Oh, no, it's good you jumped in there. Yeah, so they did say they were going to go back to like their team and see if they have BAM files, right, Stas?
Yeah.
But we haven't heard from them yet.
Right. I think let me check the last communication. I think she said she doesn't have raw data, but maybe she was also very confused when I asked about raw data, which to me feels like, before we worked with someone who was kind of more of a bioinformatician, or at least worked with raw data. So maybe, I don't know.
Wasn't that person from Guardant, Stas?
Yeah, her name was Leslie, and her last name started with a K. There were a couple Leslies we worked with. The first was
Yeah.
Leslie, and her last name was started with a K, and then there was another Leslie whose last name started
Leslie
with a
B.
B.
Yeah.
But Leslie B was more on the, I think, project management side, less on the engineering side.
Yeah, Leslie Kay, she's a genetic counselor and scientist, so she understands.
Okay, so I'm looking, this is from Nalima, and it says, hi, Lehman Stas, we do not have any raw data from Guardant. And then she links some tables that she found with some more information, which is helpful. Raw data would be ideal, but I could
Can
try
you
to
sh-
make it work.
Can you show me the email?
Yeah. All right.
Mm,
Can you see that?
Is she
Yeah.
working for Guardant or is she?
Nilima is our data steward from the Northwestern side. So she manages the data portal from Guardant on our end.
So we do not have any raw data from Guardant, but I found.
So really what's important to, at least to the intratumor heterogeneity work, is having some measure of allele frequency, which we have here. And then copy number data. But not all of their, not all of the alleles or SNVs have copy number data. So that could be a potential issue. Unless we assume that there is no copy number variability for the ones that they don't provide copy number variability for.
Huh. I mean, they don't have raw data, but they can pull this data out? I don't understand.
Right.
So it seems like this data is already organized in like their database. It's like a column that's already organized in their database that they can send to us. And I don't know, how much access they have to the raw data, or if this is just what they've received from Guardant.
Hmm.
Because I think it could be a possibility that Guardant didn't share the raw data through the portal.
Trying to understand. But last time I talked to Guardant, they said they are sharing raw data, but I guess it's just like at what level, right?
Mhm.
So Stas, for PyClone, what you need is for each variant, you need copy number and allele frequency,
Yeah.
and you need it for all like SNVs. Can you,
Correct.
what about indels?
Well, Indel's PyClone doesn't really look at.
Does not?
No, I mean,
Okay,
as far as I can tell,
right. And then amplification, no, right? Deletion, no. Yeah.
nope.
So just SNV.
I mean, amplifications, yes, kind of, if again, in that region there is a mutation. If there's a mutation and it gets amplified, then
Right.
yes. But
Yeah.
if there isn't a mutation and it's just duplication or any sort of amplification, then no.
Right. And remember you reviewed Charlie Swanton's paper before, and they calculate ITH, but how they did it is pretty similar to what you've done, right?
Yeah.
Yeah. Just like PyClone way.
The other issue here, because this is blood, there's there's less mutations in total. PyClone does need, I forget the exact number, it does need some minimum number of mutations. So that's another thing to keep in mind, something that they can't really change. If the sample has enough mutations, great, we can run it. If it doesn't, we can't. Something else just to keep in mind.
So one thing I want to clarify is, remember when we when we looked at Garden, this is like 2 years ago when
Yeah.
you and Haesung worked together on Garden data. The Garden worked with like 70 gene sets, remember? And now this is like from from that time, it's almost been like 2 years now that they are using more than 300 genes to calculate blood TMB. So if you look at Garden report, there's a TMB, and you know that you need at least 1 megabase pair length to
Right.
calculate TMB. Well, exome sequencing is best, but you need to call it TMB, you can't do that from 70 genes. So so they did call mutation based on like 300 genes from their new Garden panel. So what we're requesting from them is this 300 gene, not 70. But what they do is they report just the 70 gene mutation still. But they're sequencing 300 genes because of the TMB option.
Right.
So what we're requesting is 300, not 70. So I want you to communicate with Lima that the SMVs we get are not from 70 gene panel, but the whole panel they're using to calculate TMB. Because the number of
Right.
gene mutations matter in terms of PyClone accuracy. Does that
Yeah.
make sense?
Yeah.
Once you clarify that, I don't have problem just getting those columns and getting that report out, that the numbers out, so that you can calculate PyClone from each Garden and do the ITH analysis.
Right. I do remember we were having a conversation about the different gene panel sizes with Leslie B.
Uh-huh.
And here, I'll share my screen again. So this was back in, this was just earlier this year. So probably not much has changed. So I reached out to her about these gene panels, the different sizes. And she replied and said, the panels haven't changed. They're still using the 74 gene panel. And they do assess TMB on the backbone of 500 genes. And then I asked her if there's information about it, and she said that we don't release specific gene level alterations for our TMB. It's simply a calculation of mutations per megabase of the backbone. But you need to know if there's a mutation there, which means you need to know where it is and what the mutation is to identify it as a mutation, to calculate TMB. So they must have that data.
Right.
Yeah.
They must have that data.
But they
to
don't release it.
calculate TMB. So we don't release specific gene level alteration for our TMB. So do you think if they have that mutation data, they should have the allele frequency and copy number for it, or is it
Right? This is assuming that the reason they don't have it is due to, like, it's possible that there's a storage issue or a logistics workflow issue or a privacy, I don't know. That's also, or maybe it's a quality issue. Maybe they're not as confident in those mutations, and so they can't report them as possible SNVs with certain significance. That there might be some other reason why they don't report it. Like as far as data generation goes, they should have that data. The question is it have high enough quality? Are they able to store it? That's the only thing I can think of that would explain why they don't release it.
I see.
If that makes sense.
I see.
Maybe there's some other reason, but that's what comes to mind.
I think if we don't get it, there's no point, right? So
Yeah.
I think we have to push for this. So I still. Um. Leslie Buchheit. How about to CC everybody in Gardens, like that you've been in communication, and CC Nima.
Okay. What,
And
what about Leslie K, the one I worked with before?
yeah. CC her as well. And also CC our own rep. What's her name? Liam.
Oh, I'm blinking. Let me look it up.
Because she can help out as well. You know who she is. She
Yeah.
to arrange talks and all that. But CC everyone and just say, hey, Lima and
It's Shelly.
Shelly, Marino.
Yep.
Yeah.
Okay.
Yeah, you should have the email to Stas. If not, Liam, you can send them.
Yep.
So the point is just say that it is critical that we have access to the 900 gene backbone mutation data. And Dr. Che was told that he would have access to those mutation data. It is critical to have those mutation data in order to calculate ITH. So can you clarify that these mutation data would come from not just 70 genes or 900 genes? If not, can we make this happen?
Okay.
Yeah, I think before pulling it out, I think this is really critical. And if they say no, I'm gonna have like maybe like a Zoom call with higher up.
Okay.
I have access to CEO or like we can leverage, we can tell
Right.
them, if you're not giving it to us, we would rather work with another company who would give us ITA, like mutation
Right.
data. Now they're calculating TMB. There's so many different companies. I don't like, there's no reason for me to, work with you guys. If you're not, you have the data. It's not like, it's just a workflow. If it's just
Yeah.
a workflow issue and if you don't have it, you can't share it. Okay, we understand it. You have it, you're calculating TMB and you choose not to share it. I don't understand. I would rather find another partner who can share that.
Right.
We have to, you have to be very strong. And once so from 500 gene data, we can get more accurate ITH and we can do a lot of interesting analysis, which can benefit our patients. So I think we're gonna make that happen. So let's work on that, and then get the raw files. Okay, we're
Yeah.
almost there. And
Yeah.
once we get it, you can work with Haesung. There's Dr. Minjung's joining as another postdoc. So yeah, we're all definitely interested in ITH. And it will yeah. I think a lot of companies are working on it, and like you mentioned digital pathology, like digital
Yeah.
pathology, can we assess heterogeneity at
That
the
was
yeah. And then it has to, and then we have to show, let's see, I want to show, with the, H&E slides showing heterogeneity, and then can we match that with the clonal heterogeneity?
right. So
Yeah.
that was kind of the idea I had for this project, for this informatics elective, and the mentor that they kind of matched me up with, or like the course leader, I think he's good at enabling students to pursue these projects. And so that was kind of the line that I was going to discuss with him. Can we use nuclear morphology, cell morphology? How can we look at just the cancer cells and say, what's the variability here? And then assign some heterogeneity score, and then look at concordance and discordance.
Yeah. Did
Data.
you talk to Lee Cooper?
No? Oh yeah, I know who. No, that's a different person. I've heard that name before.
So he's another digital pathology AI guru. We have it here in Northwestern. He's a pathology department, so he's
Okay.
someone you can talk to as well.
Okay, I will keep that in mind.
Yeah.
I actually have a meeting with the course director today, so I think a
Oh,
lot
yeah.
of stuff will get clarified today and then,
Good,
you know.
because he's the one who his lab people are the ones who scans all our H&E slides.
Oh, that's
Yeah,
awesome.
yeah.
Well, TCGA has diagnostic slides and tissue slides, and they have at least 3 slides per patient, which is, to me, seems like enough data to at least evaluate something.
Yeah. Yeah. And Cooper has his own like open source like scanned H&E sites in his
Oh,
own
perfect.
cloud too. So he can tell you about it too.
Right, because that's the hard part. You need slides that have been, as far as I understand, that have been annotated to train these algorithms, which is very time consuming, but if that's already set up, that makes things a lot easier.
Yeah, and then we work with a company called Lunit, and they've
Yeah.
already, you know that they've already analyzed the H&E slide in terms of the lymphocyte, but they
Right.
are doing it in terms of, how you, let's say you, with multiplex immunofluorescence, you identify CD8 B cells, and then just train with TCA slides. Hey, these are B cells. Can you identify them? Just you with H&E slide.
Right.
How wonderful it would be, and they're almost there. The technology is far out that if you just looking at H&E with 90% accuracy, you can find ALK fusion, EGFR mutation.
Very cool.
Yeah. So why can't we do that with ITH?
Right.
So
in all the reviews that I've read on this over the last few days, it seems like kind of the 2 directions that most people are going in are TILs, and then looking at the context that the cancer cells are sitting in. How much stroma, how much fibrosis, how much necrosis, but I haven't found, I think maybe only just a couple papers where they're actually looking at nuclear morphology or cellular morphology. So this is definitely kind of more on the cutting edge as far as digital pathology goes and image analysis.
yeah, maybe that could be at the level of electron microscope, because like the cell morphology, nuclear,
Right.
H&E slide is sort of like still at the more like a high level.
Yeah, maybe that's the limitation why it hasn't been done, but I know they do look at nuclear morphology for diagnosis. So there are papers I've seen that use that information to decide cancer, not cancer. So
Right.
there are tools out there that do this. They just haven't been used in this way yet, I think.
I see.
Maybe again, there, like you said, maybe there's a technological limitation or something, but
I see. And there's a shareware people use to train H&E slides. I forgot the name. Liam, do you remember? This Gia from Jack's laboratory, she was supposed to teach us about this shareware algorithm. Anyway, I forgot, but it's most papers use this algorithm to analyze H&E unbiased way. But anyway, we can invite Stas if that happens. Liam, just remember him. So there's a question for you, Liam. So sharing that, like what the Jack's MT CDA or CDA, and sharing that Lunit that our own H&E slide with them, where are we with that?
Yeah, so initially we had requested the MTAs to Kim Matter, but she's been apparently too busy and she's not responding. So I had to reach out to Christine Miller, and I sent her all of the MTAs that we had requested to Kim, and Christine said she'll work on those, and this was last week.
Okay, so that's with that. So once that's done, then we can just upload our H&E slide de-identified ones to JAX's cloud, and they can run their program. But the point is, when they do that, we'll have our own people learn from them. And invite Stas, because he's interested in digital pathology.
Mhm.
And Dr. Chion's gonna be in Korea by then, but still invite her, see, because she has interest in there. And you and Haesung can join.
Okay.
Okay, so let's still not forget and pursue that. Okay, anything else, Stas?
No, that's it on my end. Thank you
Okay.
very much.
No problem. So finish the paper, Guardian. Let's get that done.
Yeah.
And Tempest, what's your plan? Tempest ITH.
So with Tempest, we basically, so I think last time we talked, we were investigating signatures. I was able to get Tide to work. And so the thought I was having is, if we can pick patients who are who have Tempest data, and who we know, kind of two groups. Both groups are non-small cell lung cancer, or we can pick squamous or adeno, depending on how many patients we have. One group that we know are good responders, and one group we know are poor responders, and look to see if these signatures kind of match up with reality using what we have from Tempest. And then we can also integrate RNA data for those patients too.
Okay.
That was kind of the higher level simple idea I had for the project that could get us started.
Right. Yeah. And with that, what is your plan for ITH with Tempest?
Right. So for that, we could, oh, sorry, I didn't answer that. And then integrate ITH into that as well. Because if we can show that, well, it would be interesting to show, hey, these signatures work based on molecular data. These are the responders, these are the poor responders, and then show how ITH lines up with response or no response. Is it concordant? Is it discordant? Does it have a completely different trend? That sort of deal. We just need enough patience, and I think that was the biggest issue before, is we just didn't have we have a lot of data, but of those, which patients do we have clinical information for? Which patients had IO as a first line therapy? Which of these patients also have RNA data? And so every time we added one of these constraints, we just had less patients. But yeah, so that was the plan. Look at IO response based on signatures, and then also look at ITH and see how those two overlapped. Happy to explore other ideas, or if other kind of priorities have changed, and yeah, just let me know.
Yeah. So this is what I'm thinking. There's so many papers looking at Tide-like RNA signatures, and there's companies out there. So it's like a red ocean. It's
Okay.
like so many people are doing it already, and then we can show our own signature. But how do we change the world? Like, how do we
Right.
change treatment algorithm? So and then not many people are publishing on the field of ITH. And it's still primitive,
Right.
and it's like you said, the method is not clear. And even if we show it, like, how do you integrate it, right? So
Yeah.
I think the future is, because how can you talk about ITH when you're deciding everything based on one needle point?
Yeah.
Because there's so many other things. So and then you can't do autopsy, obviously. So it has to be blood and tissue altogether.
Right. And then the slide too, potentially.
And
Because
the
I think
slide too.
that's another dimension that isn't really captured by molecular data. Just because there's a mutation doesn't necessarily mean that it has a phenotype, but on the slide you see the phenotype,
Mhm,
but
mhm.
you know,
Yeah. So, that H&E heterogeneity is a bigger question, and it's like it's gonna take a lot of time
yeah,
to, how do you train, like how do you define heterogeneity? Like what is it? And we have radiomics too, but I think we can start with what is the best way to show ITH works, and it works better if you have both blood and tissue data.
right.
So I haven't seen any paper, even now, like we talked about this several years ago when we were talking about the review paper we wrote
Yeah,
together. And I haven't seen any like blood ITH, tissue ITH analyzed together. It's almost pathetic that,
yeah,
no one's doing it. It has to be, I feel like it's something that has to be done, that no one's doing it. But that's why we want to get raw data from Tempus and Garden. That's why every new patient I see, I've been ordering both at the same time. Tempus, ideally, it has to be, it's better, it's run on a same platform. However, Tempus's blood liquid biopsy sensitivity is not up to par with Garden yet. That is the reason why. And Tempus blood TMB, I don't know if they give out TMB by blood. I don't think they do. Liam, can you look into this? Can you see the Tempus report and see if the blood liquid biopsy produces TMB these days? I haven't seen one, but you can ask anyone, like the Tempus people, if they are doing it.
So if they're reporting TMB on liquid biopsy.
Right.
Mhm.
And if they are doing it, what I am trying to do now is just reflex concurrent blood and tissue TMB, if you know what I mean. That way they're running blood and tissue in the same sequencer, Illumina. And both are generating TMB, should be enough to calculate ITH on both ends. So which was better than using Garden and Tempus. So that's kind of future plan. But what I would do is at least start with Garden and tissue Tempus ITH calculation, and try to correlate that with anything immune microenvironment, signature, survival outcome, everything. So I think for that, yeah, go
yeah,
ahead.
what I was gonna say, that's all doable. We just need to pick a patient cohort to start with. Doesn't have to be a lot of patients to start with, so we can build out kind of the workflow and the associations. Even if it's like 10 or 20 patients, we can start with responders, or we can good responders, or we can start with poor responders. But I think that having that list of patients, like, hey, let's do these patients, will be very helpful for me to kind of focus my efforts.
Yeah. So I think Haesung and you've been working together.
Mhm.
So Haesung has Tempus projects already. So it would be good to use those patients and some more additional patients for that project.
Yeah.
So I think I'll just work with Haesung to identify those patients, and then you know how to get the raw Tempus data and calculate the ITH, and we'll do the clinical annotation of of those patients in our lab, and go from there. So I'll
Yeah,
sit down
that's
with Haesung and talk about it. But
perfect. Yeah,
that
as
will
long as
be our next project, like hands-on next project. Yeah.
yeah, that sounds good to me. As long as I can get MRNs, maybe names and birthdays, just in case the MRNs are different, I should be able to do it, no problem.
Okay, very good, very good. Okay,
The Tempest data that we'll be getting will be from like multiple institutions though, so I'm
right,
not sure if the MRM will get the MRMs, I'm not sure, I think they will be completely de-identified.
right. Those data are coming from all this institution, but the one Stas will be doing will be our own. So
Oh, okay.
so we remember we have our own radiomics and our own LUNAT immunotherapy database. So it will be the combination of the LUNAT, Veristrat, and the radiomics data. And what we need to do is, the question is who has Tempest data? And
Right.
among those three database, who has Tempest data will be included in the Stas database. So I think this is a bigger task. So I think Hesong and Liam, you guys can work on generating those the immunotherapy database for Tempest. I mean, it was there before. We worked on it before. So it's, and then Dr. Jian can help us too. So this is anybody who in whether it's first line, second line, or beyond, anyone who was treated with immunotherapy who has Tempest data. Very a simple criteria. So you can utilize Veristrat data, radiomics data, LUNAT data to find these patients. And the more the better. So the question is, how does tissue ITH, intratumoral heterogeneity, affect survival outcome of patients who received immunotherapy? Very simple. And then we're going to do the same thing with Guardant. With Guardant ITH, blood TMB, blood tissue, blood ITH, and then hopefully someday we can combine both, and then come up with a better finding. But tissue is all we have right now, so I think that's low-hanging fruit. So let's identify that data. So identifying the data. So let's see. So Hesong, you're the main lead with Stas on that. Identifying the patient database, and Liam and Dr. Chiang, while you're here, can help utilize existing data to identify those patients. Let's do that.
Yeah, so I'm thinking the easiest route or path would be to just screen all of the radiomics patients, the data set patients, and see whoever has a Tempest report on it. And then I think that could be a nice initial cohort to begin with. So right now, the radiomics data set is around like 138. We're going to build that out to 200 in the future. But right now we have 138. And these are all Dr. Che's patients. I assume most of them would have Tempest reports on them anyways. So I think that will be a good place to start. So I think we can get like some of the students to help us go through the list and check for Tempest reports.
So yeah, let's start with there. So the 200 patient list does contain our thoracic group patient who, but I know some of them order Tempest, not just me. So Veristrat, I think I'm the only one who orders it, but Tempest is ordered by other physicians too. So still look for those 200 patient lists to find the Tempest, but again, the radiomics limited immunotherapy to frontline, right? Did we include second line too, or frontline?
It's primarily frontline, but
I'm pretty sure Dr. Chiang said just frontline.
Yeah.
Okay. So we can start with the the frontline 200 patients, but we could not forget the prior immunotherapy cohorts from Beristrat, from Lunit. I think Lunit's frontline too, if I'm not mistaken. Maybe it's not. I'm not too sure. But the goal is to catch all lines of immunotherapy, not just frontline. But we can start with radiomics, but don't just end there. But make sure we tap into the all other existing database.
I just want to quickly mention, I'm looking at this Tempus XT like PDF, and it says like the third version of the XT assay covers 648 genes, and it also reports TMB. And XT is usually for blood samples, and so I think it's saying it does measure blood TMB. Have you seen any of that in your patient report?
So if it says so, that it should be reporting it then. So I think we have to reflex it. But so something we can look into is, because I've been reflexing Tempus data, Tempus like blood and tissue concurrently. So we can start to collect that, because that will give us raw data file for liquid as well. But I'm still concerned about their sensitivity. It's not picking up much. But anyway, let's generate the database and hand it to and run it by me. Let's give it like a week or two to get the Tempus immunotherapy data. So Haesung, you're in charge and work with Liam and Chion. And Stas, you can focus on finishing the paper and generating ITH once you get the data.
So I think while we're screening this data set for the patients with the Tempus report, I think it might be best just to start collecting the Tempus report, like the information and data in the reports. So are there specific variables that we would like to collect from these reports as we screen these patients?
Good question. So the variables would be same as radiomics database. So just use that Excel spreadsheet.
Okay.
And we would need like the response and everything. So the RECIST response, PFS, OS, those are what we're going to look for as well.
Mm-hmm.
The lines of therapy.
And then from the Tempus reports themselves, so we're just going to collect like the gene mutation profiles and TMB reported.
Right. That's all. And yeah, we're going to work with ITH with raw file. Yeah.
Okay.
And see if there's any blood TMB data as well. Right?
Yeah. Oh, also it would be good to at least just a checkbox whether RNA sequencing was done or not. So that way we know. Because it was alarmingly low percentage that RNA was available. So okay, so let's get that launched. I know we were kind of planning on doing it, but like I think this is the right time to do it. Okay. Dr. Jian, do you want to go next?
Yeah, okay. Today might be my last lab meeting in US. I'm gonna join it later, but today. Okay, so my first project is still under review. And the second one, I already uploaded the revised version. It's been a month and I sent an email to the editor, but they're still working on, so I have to wait a little bit more. And the third was a review article. It is already published, as I told you. So this was the article.
Is that, oh, is that out? Like,
Yeah,
all-
totally out. Yeah.
can you, in our lab meeting, can you send
Okay.
the link to the article so that everyone can read?
Okay.
Yeah.
Mhm. Yeah, I see. I think it is still free, but I'm not sure. It might be changed as a paid article. Because I didn't pay anything for this article.
Oh.
Can you access?
Yeah, it's still free.
Uh-huh, okay. Do you want to share the screen?
Oh, no, I think we're good. You can continue. It's okay.
Oh, okay. So review article is published thanks to Dr. Chang. I got one during my research year. I wrote the 3, but finally I got one. Okay. And my project, another one is a radio mix. It was super interesting actually, because Professor Yuri, I think he's still learning how to the segmenting, because new skills and new techniques he keep telling us, so we had to change and change and change. I have no idea. So we're doing good, but at least we have 131 patients, and for those patients, there were 297 intrathoracic lesions.
Hmm.
So we're doing segmenting all the lesions to compare the nodules versus the lymph nodes.
Uh-huh.
So as far as I know, Professor Yuri told me they should be different if the lesion is a lymph node. So texture could be different. So we can compare. There was no article that comparing nodules versus lymph node. There was an article, but it was about breast cancer, but not lung cancer.
Hmm.
So we can compare those things, and we can compare also the responder versus non-responder, as well as IR responder versus a durable responder versus a non-responder. So
Okay,
that's why.
so let me add to that. So when I went to the lung conference in Hawaii,
Mhm.
I met a lot of different researchers doing AI radiomics and AI pathomics. And they're doing very similar things we're doing. It's not just us. But I asked them about intrathoracic lesion and what they think about it. And they said they always scan like head to toe and characterize every lesion, like with radiomics, adrenal gland, bone, everywhere. And the response, they predict response to all lesions.
Mhm.
So they don't have that kind of, they're not doing intrathoracic as we're doing. And that's probably what most people are doing.
Mhm.
But I think we have an interesting question that, if lung lesion is responding, and I guess if our only input is the lung lesions or lung lymph nodes, how accurate is it just to look at the response in that lesion? And intrathoracic response, versus extrathoracic response. How is that correlated? Just looking at that level, I think it's a really scientifically interesting question. And this goes back to Stas's question, just looking at tissue, how heterogeneous is it? So we can come up with some index like discordance between intrathoracic response and the extrathoracic response. If there's higher discordance in terms of response, then you have higher heterogeneity. And does that kind of high heterogeneity seen in intrathoracic radiomics features? So there's a lot, and then lesion by lesion, how different are these lesions looking? Because we compare the radiomic feature of each lesions, and then come up with, and then if let's say all the radiomics features are converted into numbers, and these numbers, let's say if you have 5 lesions, there's going to be some sort of distribution. And it's like the same thing Stas is doing with PyClone. It's all about the distribution and how by distribution we saw, oh, this might be same clone, this might be a different clone. So same thing, if you have different number of lesions and radiomics features are, these 2 are same, but these 3 are different but similar, we can sort of come up with some sort of heterogeneity index among the intrathoracic lesion. And that might tell us something about response. So I think as we dissect deeper, we can come up with a creative index that explains heterogeneity. And it's a heterogeneity internodule and inter lymph node, and internode in between nodule and lymph node, and intrathoracic versus extrathoracic. So we can look at it a very different angle, and then see by that how the outcome of each lesion response versus intrathoracic, extrathoracic response differ, and how does that affect long-term survival. The higher the heterogeneity may be meaning worse survival outcome. But I don't think anyone has shown that in any radiomics paper, that sort of like a heterogeneity question. It's just the radiomics feature, can it predict or not predict? It's very very simple question. But we can really delve into this and dissect question by question. So I think as we build up more data, you can do it. And I think Dr. Jian is motivated enough to take this to Korea and and then once she learned enough from breast is still in the thorax. If you
Yes, yeah,
so we're
exactly.
in the
Yeah,
same field.
yeah. Thoracic cancers,
Yes.
lung and breast.
Yeah.
Those
yeah.
are different but similar.
Yes. So we could continue to collaborate
Okay.
and learn together. Yeah, I think this is really exciting that
Yeah.
and then it's like a building infrastructure. That's what you guys did. Like, correctly annotating outcome and using the best platform. We use the image trial so that we can use it wherever you are in the world, and you using this, the resist becomes really easy once you do that. And then segmentation is, I think Dr. Yuri is still optimizing what the best way is that
Yes.
so with all this, this is a question for you. So when I read the last version of the abstract submitted to Sissy, it was very different from Chisang's abstract.
Yeah.
And
Actually, we failed to make a good value for sensitivity and specificity. The sensitivity was really good, but specificity was less than 10%. We can't make it as an abstract, but we couldn't find what was wrong. So Professor Yuri asked us to change the direction of the abstract. So it is super difficult. I can't understand at all, but maybe AI experts, they can understand what we said.
Ah,
So yeah.
so well, this is a good time. Can you pull up that abstract and share that? So I want to just go over that.
So this was the first one. Radiomics based machine learning. Oh, yeah.
Yeah, let's go to the results. So 130 patients.
Yeah, give me 1 second. It was not the one. We uploaded to the drive. Okay, Professor Yuris. So, Professor Yuri told us that we can't use term machine learning, so we had to delete it from the title.
Oh,
It
okay.
was quite different. I'm not sure, but he said at least for these two abstract, we can't use machine learning terms. So
Okay.
we changed it,
I see.
and this was the one.
Right, so let's go to results. Can you just go down?
Okay. Mhm.
So tumor size significant, statistically significant between responders, non-responders. I guess. Is it the larger, the worse,
Oh, yeah, sure.
right?
Mhm.
So that's not clear here.
Sorry, it was just a clinical data.
Yeah, and significant difference in the intercept and slope of morphological integrated intensity computed for primary tumors was found in IR versus criteria and slope difference. So this is all foreign to me. Can you explain?
Sorry. But Dr. Yuri made the research, but unfortunately I couldn't understand. I read it several times, but really I couldn't understand. Sorry.
The same radiomics feature was statistically significant between responders and non-responders per IR RECIST 1.1 criteria with the intercept and the slope with the intercept difference and slope difference. For the peritumoral space, gray level site zone matrix, large zone, low gray level emphasis demonstrated significant difference in the intercept and slope. And IR RECIST associated difference intercept was 1.9 and the slope difference was minus 0.5. So conclusion is CT-based radiation features may help predict potential responders to immunotherapy. So I feel like, I know Dr. Yuri wrote this, but maybe we should have another telecon with him for him to explain what this really means.
Mhm.
For our own learning, and then kind of figure out what the better way to write it so that the clinicians can, understand it. Not the AI researchers, but I think,
Yes, that's right. Yeah.
yeah,
Yeah, I'm sorry. We didn't have much time to revise it as for the clinicians.
yeah. So maybe while you're here, or Jian, while you're here, maybe just visit him. If he's working, if he's not working from home, like see if he's around and just
Mhm,
sit down with him and kind of he'll show you the
mhm,
table with these intercept and what the slope looks like and what it means.
mhm.
And that way, because it's always hard to find the mutual like common time.
That's right.
So
Mhm,
just anytime during this week,
mhm.
maybe tomorrow, just go and meet with him.
Yeah.
And
Peter, every day he goes to Professor Yuri, so let's get
yeah,
together. Yeah.
yeah, just go there and like, yeah, learn from him.
Mhm,
And why, and I don't know any other radiomics paper, writing results like this, because this is too technical, right?
mhm.
So yes, for method, you can be as technical as possible. For results, I think you have to
Okay.
be more interpretive. And
Mhm.
what so yeah, maybe do that while you're here this week.
Okay.
Okay, all right, just wanted to mention that. We could go back to your slide.
And another abstract.
Yeah, so looks pretty similar, right?
Yes,
And this one, same, so this one,
pneumonitis.
yeah, tumor size demonstrates. So this one was a little bit interesting because, it's this tumor. So this may be hypothesis generating too. But do you know which direction? The bigger tumor, immune toxicity, or smaller tumor?
Immune toxicity,
Yeah,
pneumonitis, you mean?
yeah.
Size versus pneumonitis. I didn't compare both them because I only saw the responder versus non-responder
Mm,
based on IR resist.
mm. So, let's go back to the first sentence of the result. So the common perception is if you develop thyroiditis, you're more likely to have response to immunotherapy.
Mhm.
Okay. But here it's the opposite. Like if you develop pneumonitis, you're more likely to be non-responder. Wait. More common in non-responder?
Yes.
But why is it 8% versus 14%? Isn't that opposite? Oh, wait.
No,
Non-responder.
non-responder is more common.
Oh, okay, I got it. It's just non-responder, but the responder is 8.8, non-responder is 4.
Mhm.
So it's kind of contrary to our common perception.
Um,
It's like Avastin has, high blood pressure group has more responses.
mhm.
Higher platelet count baseline was more prevalent in non-responders, so we know that patients who receive chemotherapy continuation were more likely to be durable responder. Okay. So two features and the tumor size. Tumor size. So ask Dr. Yuri whether
Okay.
what direction this is. Larger tumor causes more CIP,
CIP. Okay.
but I don't understand the significant difference between radiation related and CIP. So what is that difference, radiation related and CIP? Because that wasn't really the focus of our paper. Different. So I rather want to know, because we know if it's large tumor, you're going to have more radiation pneumonitis because the field gets larger. So we are not really interested in difference between radiation and CIP.
That's right. Yeah.
We're kind of only interested, does tumor size relate to risk of CIP? So maybe look into that. Significant difference in intercept and slope of the neighboring gray tone difference metrics. Coarseness computed for primary tumors was found in CIP. For peritumoral space, densities also showed significant difference in the intercept and slope. And CIP associated with difference in intercept equal and slope. So these radiomics metrics are pretty well known to radiomics specialists, I think. And it's just a way of telling that there is a significance. But you know, clinically speaking, I think you know, if you have tumor here, and then you have your background is now pneumonitis, the peritumoral, you know, qualities will change. But I guess, you know, we're predicting it before this is before start of immunotherapy, so it still makes sense. Yeah. But again, do you remember sensitivity and specificity for this one?
No, Professor Yuri didn't give us that answer.
So why don't we, just for the sake of our own learning, sensitivity, specificity, and AUC
Yes. Okay.
for both models? I mean, we need to know, we need to be convinced, not just say, you know, it will help predict, but what's the level of accuracy? We're not disclosing that here, so
Sure. Yeah.
I don't know what the reviewer is gonna think, but I think we just have to, you know, even for a poster, I feel like even if it's not in the abstract, I
Yes.
feel like in the
Yeah.
poster, we do have to disclose it. And
Yeah,
say
that's right.
it's a very limited model, you know? So yeah, we're gonna have to take it to poster if it gets accepted, so let's work on it. And, you know, we remember, Dr. Chiang, we said we still want to write this up because it will take time to get the 200 patients.
Yes.
So, you know, whether this predicts the model well, or if it, you know, doesn't, I think we can just describe our best effort and it did predict or it didn't predict and still write it up.
Yeah. Okay. And actually, Peter, Taegu, and I'm doing not only for the cancer segmenting, but also the normal lung parenchyma and the spleen, because Professor Yuri recommended to reduce the bias, which means each patient may use a different machine, and they have different texture of normal lung parenchyma and spleen, but those are very homogeneous organs. So
Oh,
we are doing together, and it's quite different way what the radiomics showed so far. So
so
yeah.
segmenting spleen
yes,
and normal lung.
yes. So that's why we're taking time. It takes
But how do you define normal lung?
normal. When you change the lung setting view, we can change all the views. So we can find from that CT scan. So I mean the lung parenchyma without cancer area.
Mm,
I can't 100% sure that that is normal, but at least that there is no cancer.
okay. Okay. Yeah, I mean, I guess the background can be COPD, it doesn't matter, just the non-tumor lung.
Yeah, that's right.
So that will help to decrease the noise.
Yes, he
Yeah.
said like that.
Okay. Mm.
Peter, can you explain a little bit more?
Yeah, so it's actually a paper that you co-wrote with him a couple of years ago on scanner-associated bias. I sent it in our group chat. And so when we segment these different tissues, they show up in a histogram live time as different intensities. And so when we're segmenting things like quote unquote like normal lung parenchyma, then it's just going to straight up show as -1000. It's going to be like a very specific range in the histogram. And so that's how Dr. Yuri wants us to track it. Same thing with the spleen as well. We're excluding any calcifications or anything like that, any vessels. And on top of that, he already has an existing database that he used for the scanner-associated bias paper he wrote with you. And so if he crunches the numbers and let's say it doesn't reduce the noise, then he said he'll just add on his own data set from the previous study done to eliminate scanner-associated bias. And he said that that combined with new segmentation techniques that we've learned, he said it should reduce the noise pretty substantially. So we're pretty hopeful that that'll probably uncover more like significant radiomics features that would help us better predict outcome.
Okay. Okay. Yeah. So how long does that take?
I don't know.
Maybe Wednesday we can finish up. I really want to see that research before my leaving. That's why I expanded 1 more week. So
Yeah, we'll
I
we'll
hope.
do it as quick as possible. We're
Mhm.
gonna try to. We have around like 115, 120 lesions left to segment, and so we're gonna try to finish by Wednesday or Thursday, and then hopefully Dr. Yuri can get started on analysis this week as well.
Okay. Okay. Yeah, I feel like we're learning as we, and a lot of it is very technical, but
Mhm.
yeah, as long as we understand the concept. And I think that's why, I mean, that's why I like to work with Dr. Yuri, but also at the same time work with other people who have different algorithm, different thoughts. Some people are actually opposed to segmenting. Some people are just like unbiased, let the machine figure out what's important, what's
Okay.
not important.
Mhm.
But at the same time, I don't think machine knows what's tumor.
No,
No, there's no way.
no way.
No, it doesn't. It doesn't because
Even Professor Yuri, he couldn't find the exact cancer.
and
Actually,
there's
some
like
cases.
there's like formulaic functions on LifeX that you can like exclude a certain range, like it make a threshold. for the machine to learn, but then it'll include like blood vessels and all of these other things. And so that's why even though we have these things like end
exclude,
function
yes, we have.
that exclude it automatically, we actually have to go in and erase like all the blood vessels
Yes.
manually. And so I don't think there's a way for machine to do that yet.
Mm, yeah.
AI can't replace human. Can't. Impossible.
Yeah, yeah, very interesting. Okay, very good. So hopefully
Mhm.
it will improve. So before we get to other slide, I'm gonna ask Liam. So where are we with the there's a picture health and there's one more company, right? What is it? Image AI? Was that image?
Yes.
Yeah, it was a trial.
Image
Mhm.
Image trial is a program. Image was there
AI.
image?
So there's three. So one is image trials. We're continuing to upload those. I believe Will is working on them right now.
Only 20 cases left. I think we can finish up today.
Okay, so that's image trials. Picture health, Dr. Hesung is in charge, but we have successfully sent them all of our DICOM files.
Mhm.
We're going to be sending them our segmentations once we finish segmenting, either tomorrow or Thursday.
Mhm.
Then we have also received the H&E slides from Dr. Isel for the ones that we already have from our project with Lunit.
Uh-huh.
That is ready to be shared. And then the other DICOM files we've requested to the hospital. So they said that might take about a month or so. So we're waiting on those. And then we are currently also in the process of identifying the slide numbers for all the other H&E slides in that data set. So once we are able to identify and collect all the slide numbers, then we can request those to Dr. Alexiev again, so he can pull the slides for us. And then there is another company called Imagine AI,
Right.
which we have the MTA for, and we haven't communicated with them since then. So I think it would be a good time to touch base with them and see what they would like. But I think we can basically use the same process we have that we use to share our data with Picture Health, and we can just share it to Imagine AI as well.
Yeah. Remind me, is this company the both digital path
Yes.
and yeah, this does both, right?
Mhm.
So I think it's still yeah, I don't think we need to do any Zoom meeting yet. Just email them and ask them, we have MTA in place. What's the best way to send you both H&E slide and DICOM files? And just do the same thing you've done with Picture Health. And once they receive it, let's do a Zoom meeting.
Okay.
And with Picture Health, I think we're getting some support. So maybe with Imagine, I don't know, we haven't discussed any support from them, right?
We brought it up. It was a discussion that was had, and it was kind of early on in our conversation, in our discussion. So we didn't go anywhere concrete with it. But then they were definitely open to supporting publication fees and like travel for conferences and things like that.
Okay. Okay. So let's send our figure out, so more it's logistics, figure out how they want this to be shared.
Mhm.
So work on that, Liam.
Okay.
And then we can have like the follow-up Zoom call once some things are figured out.
Mhm.
So yeah. About Dr. Alex, pathologist, I know we've requested more.
Mhm.
Is that for the Lunit project as well as Picture Health, both? Or
Yeah, so we should be able to use it for all of them, because now we're working with like a centralized data set that will be able to be used for these various projects. So yes, once we receive them, we can definitely send them to Lunit as well as Picture Health and even Imagine AI. Yeah.
right. So these are kind of extension of our first line immunotherapy patient who has H&E slide.
Yes.
So we gave the list to Dr. Alex and he's working on it.
Yes.
Okay.
And we're just going to kind of help him speed up the process by identifying the individual slide numbers, like the ascension numbers. Because that will help him, identify the slides. Because we've been having some issues when we've sent him MRNs in the past.
Oh, I see.
Yeah. So according to them, the best way is to send them these accession numbers, like slide numbers.
I see. So we sent both?
No, so we sent the MRNs, but now we're collecting the ascension numbers and we'll be sending them and by end of this week. So that one,
Who's helping with that?
Will and Monica is helping us with that.
Okay, good, great.
Yeah, I asked all the students, Will, Monica, Jessica, and Grace. I've made a separate spreadsheet.
Okay, good. That's really important. So, okay, let's go back to Dr. Chian's slide.
Okay, next to look at biopsy is just like it's still in under review. So
Under review by +1 or
hey, Leon.
Leon? Oh, okay.
Mhm. And the last one is the ctDNA. Actually, yesterday we submitted to San Antonio Breast Cancer Symposium.
When's the deadline?
Yesterday, it was yesterday.
Yes, sir.
Yeah.
Are you
So
going?
I don't think so. I'm not sure because it's too far. Yeah.
You've been there before, right?
Yes, but there is nothing to visit or to play. I had to fully study. I don't want to go there again. And usually they release everything after conference, after symposium. So I can see it from online. So I don't know if they may stop to releasing. Because everyone wants to see it online, but they don't want to go to San Antonio. But isn't that good for the symposium? So most of the conference and symposium, they stop releasing the lectures after during the symposium. They go to the offline only, not online. That is really not good for the conference.
Oh, okay.
Yeah.
You know, the San Antonio, I went there a couple of times, and this is the only conference that will print your poster for free.
What?
For free.
Poster
Yeah.
was free?
If you send your poster to San Antonio, the convention center, they will print the poster.
Oh, yeah, they did it. Yeah.
On that site.
Yeah. Yes, that's right.
It's the only conference that does it. This is
Oh, yeah. I think they're rich.
yeah, and then yeah, I think they want to attract more people that way.
That's right.
But anyway, can you go over that then?
At that abstract?
Yeah.
Okay.
So any update from Predison?
Oh, not yet. They said they are still working on the tissue and methylation, but we could make the abstract, the research that only we have so far.
Okay. So background, despite the availability of effective therapies as effective therapies as an adjuvant treatment, such as TDM1, neratinib, PCRs, strongly predict survival. This study aimed to investigate the role of ctDNA analysis in predicting PCR and clinical outcome. Okay.
This actually Sujong wrote it. This one.
Total of
I didn't
34
touch anything.
underwent neoadjuvant chemotherapy combined with dual anti-HER2 agent and surgical resection were enrolled. So dual means pertuzumab
Yes.
and trastuzumab?
Herceptin, yes.
Okay.
Trastuzumab and pertuzumab.
Including 22 clinical stage 2 and 3 patients. 4 time points. Whole exome sequencing also. In evaluating blood copy number burden, genomic alteration, mutation signature. Oh, I guess IP yielded negative result, targeted NGS sequencing was conducted. Not sure what this meant.
That's that the blood samples of a baseline. If the low path showed negative, we did a beacon. The testing.
Beacon?
Yeah, we performed the beacon.
I thought that was the opposite. When low pass whole genome sequencing was positive. Remember, if it was below the 5.5 or 5.6 cutoff, those were the ones that are selected for targeted sequencing. Beacon.
Oh, care. I mean,
Care.
when it was a negative, they performed the care.
Oh, care.
And when it was a positive, they performed the whole exome sequencing. It might be beacon.
So that message is not clear because both are
Mhm.
targeted sequencing too.
Yes.
So there has to be, so when you revise this,
Mhm.
so I think it's best that it come from Paradism people. So
Okay.
they know the terminology.
Mhm.
But when IPWGS is positive, then we use beacon, and there's gotta be a, how many gene set based, serial, blood-based, MRD assessment or something like that. That's the beacon. When IPWGS was negative, then targeted NGS panel. And then in the method, you can put beacon and care as like a trademark
Mhm.
with R on the top right corner.
Mhm.
Kind of differentiate, and it was just done one time at baseline, right? Care was just done in baseline.
Mhm.
Versus beacon was done at every MRD mark. So I think the method has to be more clear.
Okay.
And here, like when it yielded negative results or something like that. Okay, and results among 34, 61% had nodal mets, 64% hormone receptor positive, 26 patients with 76% PCR positive CTA was detected in 20 patients. Higher nodal stage was significantly associated with ctDNA positivity at baseline. So I would put p-value, like what was the p-value
Mhm.
here in that 2x2 table after or like percentage
Mhm.
p, no nodes nodal, says higher nodal stage, meaning what, like N1, 2, 3, like what is like more in-depth numbers. After surgery, MRD positivity was observed in 3 patients. Among 26 patients who achieved PCR, only 1 patient showed MRD positivity, while 2 non-PCR patient demonstrated MRD positivity. So 25% versus 3.8. I guess that's the big difference.
Mhm.
So maybe p-value here as well. It's a 2x2
Mhm.
table, right?
Okay.
The presence of MRD was significantly associated with non-PCR status. The presence of ctDNA at baseline was associated with higher nodal stage, suggesting its potential as a prognostic marker. Further studies with larger cohorts are warranted to validate these findings and explore the utility. Okay. So here we're not utilizing any of dynamic, right? Yeah, what
No.
was cleared was,
Mhm.
whether clearance, it doesn't, what it does not address is the clearance of ctDNA with chemo or surgery. And how that associated with outcome. Maybe it wasn't associated, maybe that's why it wasn't mentioned. But in the manuscript, it just has to be fully discussed.
Mhm. Mhm.
Because people look at clearance rate because we're looking
Mhm.
at neoadjuvant, that's what they're looking for. If it was positive, has it been cleared by either chemo or surgery?
Mhm.
And did that matter?
Mhm.
How did those patients do?
Mhm.
And none of that's described here.
Mhm.
Maybe because it wasn't as good of an association.
Yeah.
But still, that's what the reviewers are expecting to see.
Mhm.
But again, with methylation data, we will have more to write. So
Mhm.
I think it's a good start.
Yes, and we have a plan to get the more blood samples from those 34 patients when they visit the hospital. So, yeah, I think we can. They're still in surveillance. It's been only 2 years from treatment.
Okay. Good. It's just taking more time than we thought, but
Yes, but
Uh,
because the ctDNA is pretty popular in researchers, so we need some more significant, more powerful impact.
yeah, good. So how did you arrange the authors?
Authors for abstract or authors for article?
Abstract.
Abstract, well, maybe we have to find some presenter who will visit the San Antonio. So, I
So
need
is
to
Dr.
check.
Sujong going?
I don't think so. Yeah, she came to ASCO, so I think we can't coming back to US. The hospital doesn't allow twice, in a year.
So do they have e-poster?
Oh, yeah, they have e-poster, but with the e-poster, we can't come to US. they don't support. And my hospital doesn't support.
Oh, but you can still present not going. You know, e-poster can be presented, you don't have to go, e-poster.
Yeah, that's why they don't support the grant to go to conference. So
Right, right.
usually when I,
Yeah, yes. But you don't have to go, just present e-poster. Just
yeah, that's right.
but if no one's going, I think we can withdraw and just present the better data.
No. Dr. Che in Korea,
Oh, he's
he's
going
going. Yeah, he's
to
going.
San Antonio?
Yeah.
Oh, okay. So you can just change the presenter to him if he wants to present this.
Uh-huh. Okay.
But he's but I guess the author wise, you and Sujong has to be the first and second, like the, and then he can be Dr. Che from Korea could be in front of me, my name. But you could just change the presenter to him if you know none of us are going. And with even updated data, we could have another poster if it takes longer time. Yeah, I think that's fine.
I'm gonna come back ASCO.
And then come back ASCO.
Okay. Yeah,
'Cause it
ASCO
takes
is
time.
more interesting
Mm.
than San Antonio, actually. Let's say ASCO is a conference, but San Antonio is a symposium. They usually teach. We
Mm.
don't discuss much.
Oh, okay.
So that is a big difference. Yeah.
When you come back, are you coming back with your children?
No, I don't think so. I need my time. Yeah.
So it will be good to see you in Chicago. Free
Yes,
at night.
yeah. Yeah. Oh my God. Yes. I can have my free night after 2 weeks. Okay.
Good. So yeah. So I like, you know, where we are. So let's keep working on this. This is sort of the main project.
Mhm.
And with this, Liam, so we wanted to keep following up with the long neoadjuvant. So remember we were kind of building that database. And I think Lauren was it Lauren or
Sam?
Sam, yeah, was starting on it, and then she's doing residency right now, right? Samantha.
Yeah, she should have started.
Yeah, so do you know where we are in terms of getting that patient list?
No, so I've sent her a couple of emails in the past few months, but I haven't heard from her at all.
So what is the best way? Because I think we got a list from Dr. Barak, remember?
Yes.
And did Sam say that wasn't useful, or?
No, so she was going to combine it with like this other data set she was getting through another friend of hers who was in a different lab.
Huh?
And then she's been MIA. I haven't heard anything.
Okay. Okay. Oh yeah. So before we get to there, so that's a very important one. But Dr. I feel like so Dr. Jian mentor Monica. Is Monica not joining today?
Oh, she's there. Sorry.
Oh, can she come into the screen? Does she have a laptop or
Monica, could you come here?
was she listening to this? Or she's doing on her own?
Wait, is it here? There's Monica.
Hey, hi, Monica. So you guys can be on the same screen.
Hi, good morning, everyone.
Good morning. So yeah, Monica, make sure you listen to the lab meeting and understand what's going on. But while Jian is here, this is her last week here. Learn from her about the radiomics. You can learn the technical aspect from Peter as well. But I think you'll be the main point of contact for radiomics projects. While Jian is leaving, she'll be still be involved, but we want someone to be in charge of the radiomics project here, and you are medical doctor, so we have many students, but the clinical wisdom is what we need here. Okay, so work with all the other students to make the radiomics project happen. But also I think, well, I guess the breast project, nothing's done in our lab. It's the companies over there, and the breast cancer patient data is in Korea, so it's not relevant. However, the whole concept of the neoadjuvant and the PCR and what is the predictor of response is a really valid question in lung cancer. So as a medical doctor, I want you to be involved in this project as well. So you could and then Dr. Jian can sort of explain about what the neoadjuvant is, why this is important. But the predictor of response in this project is, are there enough lymph nodes in the tissue, in a diagnostic tissue? And this was assessed by a company called Lunit, by Lunitscope. And we're still not giving up on this, Dr. Jian, to see if we can use any tissue. But we have the H&E slide, look at the lymphocyte density. And then we have radiomics. We have H&E slide, and we have the genomics of these patients. And then we have endpoint of PCR. So Stas, this is also relevant. We're not just going to do metastatic here. Now we're still doing so much of neoadjuvant that now we can look at the immunotherapy containing regimen in the neoadjuvant, and our endpoint is pretty clear: PCR rate, or we have MPR, major pathological response rate. So study all these terminology, learn what they are. So this is a simple correlation. How does the imaging trait of H&E slide, radiomics, genomics, and then we do have ctDNA when we do neoadjuvant therapy. I do check Guardant, so there is Guardant there. I just don't check it serially, so I'm not checking any, but at least we have Guardant as a baseline ctDNA for all our lung cancers. So the number one, so this is laying the foundation. What is the total list of our neoadjuvant lung cancer data? So what is the best way to look into this? So talk to Dr. Lee So Kim, because he has found a guy who did residency training here in Northwestern, but works in St. Francis. And he's helping Dr. Iser build a database because he knows how to work with, what is it called, Liam? The search engine?
EDW.
Yeah, EDW. a lot of times we have to pay biostatistician to get a data, and then we can't work real time. It's just so time consuming waiting period. But if you know how to do it, you can just extract, knowing the right ICD code for each. And so Dr. Iser's doing that. So talk to her about that method. And Liam, you can send her Dr. Barat's list. And Dr. Jian also has worked on, how to identify, looking at a big, new patient, and then what they, did they get neoadjuvant? So she built some more radiomics data that way. So work with Liam, Dr. Jian to figure out, and Iser Kim to figure out what the best way to come up with the neoadjuvant patient data in lung cancer. So it's very simple. So you have to understand what the neoadjuvant regimen is, and anyone who received neoadjuvant, they're not stage 4. This is a therapy given with surgery in mind. So come up with that patient list. So this would be for STAS, for ITH, and for HESON would be a separate patient group. So you have 2 groups of patients. Both groups will have immunotherapy received, but one is neoadjuvant chemo followed by surgery, endpoint being PCR and event-free survival and overall survival. The other is stage 4 group, or unresectable stage 3, who received immunotherapy as a palliative regimen. So with that in mind, we're gonna, for you, Monica, it will be the neoadjuvant group. So Samantha would be, is a resident who used to work on this. I think she's really busy. So you could be the lead of the neoadjuvant project, and work on the patient list, then work on getting all of those components, H&E slide, radiomics, ITH, work with STAS on ITH, and then Guardant, work with Liam and HESON on the Guardant, what's available, what's not available. And this will require a lot of data gathering, cleaning, all that, and talk to Liam on, how to work with students and assign works and all that. But this definitely needs medical doctor clinical wisdom to see who's eligible, who's not eligible, and all that. So let's actually launch that project. And it didn't go anywhere with Samantha being busy. So I think the neoadjuvant lung is really important. And you can, Jian, you can mentor Monica on this project, and you'll learn from this too, and apply to breast cancer later on. But whatever you learn from breast neoadjuvant world, you can teach her about why this is important and all that. So okay, very good. Let's go to HESON.
Yeah, I'm really excited to show you all the DART papers. Would you like me to print them out, or would you like to see it right now? For the 6 cohorts, I worked together with Liam, and Grace also helped out significantly. So it's ready for review.
Okay, for the six cohort, you were able to. So let's see what the best way is. Yeah, you can still share this. Well, yeah. Do you have it in the Google Doc?
I do. I have those, and I also have the thyroid with the updated translational components.
Awesome. So let's do the thyroid first.
Okay.
Yeah, let's share that slide.
Actually, before that, I wanted to quickly ask Liam. When I contacted Alexia, maybe last month, she said he's working on a different project with Dr. Che. Do you know which one this is? Is it the LUNAT though?
This was last month.
Yeah.
Yeah, so this probably was, no, I think he was referring to our ACC cohort that Richard was working on.
Oh, okay.
Yeah, so those slides we actually received and we scanned them. Oh, and so Dr. Chi, did you actually receive an email from Dr. Cooper regarding these slides?
About what?
So we scanned the ACC cohort that Richard was writing up.
Mm-hmm.
So Dr. Alexiev was able to retrieve those slides from our patients.
Okay.
So we have 5 more slides and then plus 1 from the outside hospital. So we should have a total of 6 slides which got scanned.
Mm-hmm.
And they said that Dr. Cooper will send you an email with like the link to the slides. I haven't received anything, so I was wondering if they had sent it to you directly.
Okay. I mean, June 28th. I have. Oh. Jessica is busy. Just work with your pathologist to have them delivered. June
Yes.
28th.
Yeah, so we already worked with Jessica. We got them all to Jessica and they're all scanned. So I guess you didn't receive anything from Dr. Cooper then.
So that was the June 28th was the last email I see. Understood. We will work with Dr. Alexiev and figure out.
Okay, yeah. So the week after, Dr. Alexiev was able to give the slides over to Jessica, and then I went over to Jessica to scan the outside hospital with her. So it should be in the system. I think I can just email Dr. Cooper asking for those slides.
And you got the one from March that says it was shared with Ether, March 29th. B. Cooper, you.
Yes, that one I believe you already have.
So it says they should not be accessed from a computer that is not administrated by Feinberg. I revoke this access.
Yeah, so Dr. Isol would have had to come in into the lab.
Into the lab and did it, yeah. So anyway, I did not get any
Understood.
but just then just follow up to see if you can get the image. Okay. Yeah, so I guess getting the Lunit slide is priority. So because we got the ACC done by Alex, then you could say this is either, just follow up with Dr. Alex. So, right. So Haesung, what are we waiting for?
Oh,
on your
okay.
end?
Wait, sorry, what was that?
On your end, the slide, that
Oh,
email.
the slides. Yeah, that's for the Picture Health and Future Lunit collaboration. It excludes the duplicates, which is like 50 here. Let me just show you this. This is what I'm talking about. So we have 209 radiomic patients, but we have the DICOM files of 138 patients. And among them, 55 of those patients already have the H&E slides scanned. And so we will send those to Picture Health. The rest of them, actually, I'm not getting the math right, but here, okay, 138 plus 55. So these 138 patients don't have their H&Es. We need to acquire the H&E slides of these 138 patients, and so I have asked 4 students, Monica, Will, Jessica, Grace, to help collect these data so that we can obtain these H&E slides from Alexia. Okay.
Oh, okay. Okay. So
Yeah,
once we,
sorry, I got the numbers confused, but yeah.
yeah, we can tell Dr. Cooper. Yeah, what, you know, what we were asking and what it contains and all that, yeah.
Okay, so this is the thyroid paper. I copied and pasted the title and the author information from the abstract that we recently wrote.
Okay.
Is that okay? This is
Yeah.
the previous version.
Okay. So, yeah, previous version is just the clinical investigators, right?
Right, yeah. All of them are somewhere, you know, in this list.
Except for a couple of physicians who moved on to corporate.
Oh, okay, yeah. So, okay, that's good enough, yeah.
Okay, and you're okay with the title as well, right?
Of the actual paper. Yes, I'm okay.
These were added to the abstract, and I think it still applies here, so we still it here as well.
Yes.
So, and Dr. Che, do you want to include the contribution? So under the acknowledgements, like the MIF biostatistics, that section all the way down to the CIDC data curation and leadership, should that be in there, or should we remove it?
So I don't know where else to put that, you know? I think it's fine.
But I believe most of these people aren't these people in the author list?
It,
Well, not all of them.
not all of them. So if they're in the author list, then we should take it out. Because it's double acknowledgement.
What about are we including Grace?
Who?
Grace Kang.
Not for this one.
Oh, okay.
She helped with any of this paper?
Not on the thyroid one, as far as
Oh, sorry, this was not that. Yeah, it was the other papers. Yeah, not
Oh.
the thyroid.
Yeah, it's okay to acknowledge students if they help with writing in the acknowledgement, you know, like manuscript writing, you know.
And then the translational relevance, I kept everything else. I just added that the dual expressing tails correlated with good outcome.
Translational relevance. So this is more like, you know, in your day-to-day clinic, how can this be translated? Not like summary. So I would say immunotherapy is not a standard of care in thyroid cancer world. And whether and data is still limited. So this data adds to the existing data that the immunotherapy for can be useful for certain patients, and the biomarker has to be a biomarker analysis helps to identify who they can be. who they will be, something like that. It's like more like a story. Then summary, yeah.
Abstract, I largely copy and pasted from the apps or yeah, from the release of the abstract. I just added that the duration of response over 2 years, it included the anaplastic patients. And then just, you know, prospective studies and various histologies.
Yeah. Very
so it's
thorough.
just-
Yeah, that's good. Yeah.
Introduction, I left it as it was, and then I added the translational component, very similar to abstract, but I think I added a bit more. And maybe it's
So remember the first version that Kara Haymaker gave us?
right. That's the one I used here.
Yeah, yes, the full version. Yeah,
Right. Yes.
that's good.
Okay. And then results, I added more information here, including the mutation status.
Mm.
And I'll write out the acronyms as well.
So what's missing is so we have many tables and figures,
Oh
supplement.
yeah,
So you just need to add all of those into each sentences.
yes. I've added those figures and I will just cite them here.
Yeah.
That's something I forgot to do.
uh yeah, I think it's pretty straightforward. Okay.
Okay. And then discussion section, would you like me to write it this way? Like create a separate, you know, like section? Or just integrate it in there, somewhere in there?
But do we have subtitle like this for others? In this,
For results, you know, methods,
Yeah.
yes, but not for discussion.
Right. For discussion, you can let go.
Maybe I'll write this
No, you
before
why did
toxicity.
why did you you got the whole paragraph deleted?
Yeah, I was gonna paste it somewhere. Oh, I thought you said
Yeah. So yeah. So in summary, so you need that in a different location. Is that what you mean?
yeah.
Oh, okay.
Yeah, so maybe we can write this like before toxicity.
Right. Wait. After toxicity, because it's all clinical.
Oh, okay. Right. Okay.
So let's look into that portion. First group, we are the first group to demonstrate the prognostic indication. I think this is really strong. It's not like this is still hypothesis generating. So we just said we report for the first time that and it's CD3 CD8 double positive, you know. I think we can just say TIL because they showed it both TIL and CD8 positive TIL, right? So it's okay to just say TIL. That the presence of TILs in this is in thyroid cancer, TILs in thyroid cancer correlated with favorable survival outcome. I want to say in patients who received immunotherapy. So yeah, take out the prognostic implication. Yeah, just yeah, just
I'll fix it and just say it led to
yeah,
favorable
just
outcomes. And
facts. We're the
Okay.
first to find that relationship.
right.
But whether it, it is, if it can be, if this is a, thyroid unique phenomenon, or, whether it's applicable in other cohorts, we don't know. It's not been validated, right? And it's very small number. So that's why we don't want to say, well, we found the truth, like, you know what I
Okay.
mean?
I got too excited.
First, observation we found, yeah. So the single expression, okay, it's study 123. Presence of PD-L1 expression, TIL tumor associated with improved PFS. Further PD-1 expression weekly with TMB showed. Expression correlated weekly with TMB showed a significant correlation with when categorized according to the following cutoffs. How additionally PD-1 positivity was linked to enrichment of pathologic variants in genes, particularly p53. So just to emphasize, when we say PD-1 expression, we're always thinking where. So PD-1 expression in TIL. Same thing. Additionally, PD-1 positivity in TIL. Just emphasize in every
Okay, I see.
sentence.
Oh, okay. Okay. Yeah.
So here, so go back to this reference, 123 patients with solid tumor. Because it analyzed 8,706 tumor samples, right? So did they
Yeah.
get PD-1 from tumor? It just reads it that way. You know what I mean?
All the tumor specimens.
Okay, so the tumor specimen means tumor and tumor microenvironment that contains TILs, right?
Right.
So the TIL PD-1 positivity was associated with longer progression-free survival and overall survival. And this was what cancer?
I feel like this was various solid tumors. Yeah, so we have like lung cancer, head and neck, blah 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 blah.
Oh,
Very systemic.
but I guess lung is 26%. Okay. Very interesting. Oh, who's the author?
Bevins.
But it's
From
Kurdra.
CST. Oh yeah, it is. Oh,
Yeah, because she likes
she
PD-1,
recommended it.
PD-1 TIL. Journal. So what is the impact factor of this journal? I'm not familiar with this. Can you look it up?
Oh, oops.
Journal of Immunotherapy and Precision Oncology.
One second.
0.77. Wow, so it's a pretty new, I guess, journal. Okay, go back. So, tumor samples for TIL PD-1 expression. Okay. Okay, so this is what Kurdrak likes. The prognostic value of till PDL1 status has been evaluated. So just say in tumor samples, various samples of various histology or some, it's not lung cancer, include or maybe including, like including lung, head and neck. Maybe you can write top 3 solid tumors of different histology or something with various different histology.
And then maybe is that too weird? Something like that.
Yeah, and head and neck and whatever was there. Just put, yeah.
Okay. I'll write maybe like 3,
Yeah,
the 3 biggest cohort. Okay.
yeah.
But this was head and neck.
Mhm. Okay, that's okay.
And then table 1, baseline characteristics, 2, best response, 3, I'll fix the table. Actually, I wanted to ask you, no, I'll ask you later. And then figure 1 is the waterfall plots, 2, swimmers plots, 3, survival. And then here, the PDL1 expression. Would you like to keep this one? I can cite it in the methods. The result.
Mm-hmm.
And then this is the uncle plot, the somatic landscape of this cohort.
Yeah, we can keep this.
Then this is a heat map analysis.
Yes. Yeah. So I don't like STA here. So we can, because others. Let's leave it for now. I mean,
Yeah, I think also response 2.2. Oh, I actually defined it. Okay. Yeah, response 2.2. I also mentioned it below in the supplemental table, but I guess we'll leave it like that. Yeah, because it's a different definition. And then not finally, it's oh, there's another one. This one maybe should go in the front, right? The front of all the translational figures.
right, because it shows what's been done.
Yeah, also this one as well, right?
Right. So, did we decide what journal?
Actually, no, I don't think so.
Weren't we aiming for like a JCO or JCO-PO?
JCO, let's aim for JCO. So JCO is a daunting task, impact factor over 40. But it has translational components, so we could try definitely. so let's figure out what the, figure, normal figure, limitation, all that. And we should figure out what goes. And my preference is everything you showed me from translational should be included in the main figure. Now, if you can't all fit it in, you know the tactics, like figure 5 A, B, C, D, E, F. So we're gonna try to fit that in.
Okay.
And then for the clinical paper, if we have to do combine the clinical figures, then we do it as A, B, C, D. So yeah, we're gonna make it happen. Just figure that out and then get my final approval.
Okay.
I think yeah. And then show me the last conclusion, because I
This one. I
yeah,
should
so
have-
in summary, you have to put like one sentence about the translational
Right.
thing in summary, okay?
Right.
Um
The favorable outcome thing with the dual
Yeah.
question. I'll do that, and then I'll figure out the figure numbers and then show you the final draft.
yeah, yeah. And you can get help from students on changing it according to the JCO format. And then we can acknowledge whoever the student is doing it too.
Okay, so that was thyroid. This is desmoid, and we've decided to submit it to JRTC short report. So I wanted to ask, last time you said you wanted your research grants and everything, your disclosures included.
Mhm.
Dr. Kurzbach has already been written. What about Dr. Patel?
Oh, that just should be taken care of by Patricia in the SWOT.
Oh, okay.
So Dr. Chi, are we gonna be submitting? We are gonna be submitting from our end, right?
Right, but we have to submit to Patricia and get
Yeah,
her
circulate it to the authors.
circulated, and then she gets of the final approval, then we submit it.
Right. And for the COIs, do we need it for both like all three co-PIs, or should it just be of Dr. Kurzrock?
Co-PIs. What do you mean co-PI?
So like the COI, should we include just Dr. Kurzrock or for you and Dr. Patel as well?
All three.
Okay.
But let Patricia do it. Okay.
Okay.
Oh, and then just ask Patricia for me on my behalf because these papers are going to be like pure SWOG papers. So ask her if there are any mechanism of SWOG supporting the publication fees
Okay.
as opposed to my personal grant, right? So it's a SWOG trial, so they should have the grant for this. Okay. Yeah, so
Yeah.
update on.
Yeah, so here last time you said talk about like the survival outcomes of patients who are refractory to the current treatments. And I was not able to find anything like that. I just only found like the response rates can vary widely from 3% to 54%. So I just want to get the point.
Okay, whatever is in the literature is all we have. Yeah, okay.
And then, yeah, patients and methods, kind of standard stuff we've seen. I updated everything. Or I mean, we as a team did. And then results, discussion. Oh, here I updated and said that although the cause of tumor regression is uncertain and could be due to spontaneous regression linked to menopausal transition or postpartum status, the fact that the two other cases of PR are male provides evidence supporting the hypothesis that the response to ICI is the underlying cause.
I like it.
Okay, thank you. Right, so that's that. And then tables and figures, baseline characteristics, best response, adverse effects, waterfall, swimmer, FSOS, and then for supplemental, more detailed.
Uh-huh.
Uh,
Yeah, pretty, yeah, obvious.
yeah. Are you okay with this paper?
Mhm.
Thank you.
So is this ready to be sent then?
This one should be, yes.
Okay. Okay, then let's so Liam, the protocol is Dr. Kurdrak has to approve it first, right? And then it goes to Patricia, right?
Yes.
Yeah. So Liam, why don't you just submit to Kurdrak, CC Patricia, myself, Megan, and Patel, because we're like the equal, yeah.
Sure.
So just say on
Move
behalf of Dr. Che, we're we're submitting this to SWOG for your approval to circulate to other authors or something like that, and then please review and let me know if you have any let us know if you have any comments. But I think you can still yeah, put Megan. It's not just to Kurdrak. Let's just say let's just say dear core authors, or just say, or just say dear Dr. Kurdrak and then just CC other people.
Mhm.
Yeah, that's better. And then yeah, just use the final really clean version once Haesung is done. Yeah. And did anyone help you with this, Haesung?
This one, Grace Kang helped out with the tables and figures.
Okay, so you can acknowledge her and then
Oh, and also with numbers. But yeah, so
yeah. Okay.
I guess something. Something like that. I don't know.
Yeah.
Okay. Yeah, I'll write this for other 5 cohorts as well. Grace really helped out a lot.
And look at the acknowledgement section,
For other papers.
other papers and see if there's any general sometimes you can write for clinical trial. We thank patients and their family members in participating in clinical trial or something. You can kind of look at other papers and see what they did.
Do we also write this? This is the thyroid paper. It mentions the financial scientific support.
Yeah, so it may be a little bit different for each cohort, so we have to get that from
Oh.
Patricia.
Okay,
Yeah,
okay,
Get
yeah,
that from Patricia.
okay. This is bronchoalveolar carcinoma.
Yeah.
This one we agreed on JTO for report. And then second is JITC.
Okay.
Should be the same. Methods, results. Discussion, no changes. It's really the same thing. I think this is also ready. Oh, except that it's just very long. The adverse effects section is very long. And so I'm wondering if we can condense this to like the laboratory abnormality for these ones, maybe. I'm not sure.
So this one, blood lymphatic system disorder, other specify. Total protein decreased. So you have to write, take out all that blood lymphatic system disorder, others, all that. Just leave the lab values, BUN, creatinine, low hematocrit.
Oh,
I don't know what TP decreases. Is that total protein?
oh, this. Oh yeah, this is total protein. P3 is thyroid, but
You have
TP
low
is total protein.
There's low total protein up. So I don't know if those are same, not sure.
Total protein should include like antibodies and etc. But albumin is just albumin.
Because like if you look at the third line that's highlighted, it actually says low total protein.
Oh, here. Oh, good point. Okay, then we'll need to check.
So this is like the raw file, right, Liam? This is what,
Yes. This is what Megan and Gabby sent over.
so right now, this is all sort of mixed up. So let's categorize it into symptoms and then laboratory abnormalities. And then in the laboratory abnormalities, like the CBC and CMP, presented in that order. You know what I mean? There's hematocrit, hemoglobin, like it's all over, mixed up.
But then create a separate category for immune mediated stuff, right? As written below here.
Right.
But I guess do something similar like symptoms, lab.
This one, I know symptom is rash and diarrhea, but you don't have to subcategorize it, but put that in that order. Symptoms
Oh, okay.
and then labs next in the under the immune mediated. And then also get rid of all the categories. Okay, so skin, like
Oh, okay. Yeah.
and just be specific. Yeah.
So you're saying don't like just like don't write symptoms, lab, just put them in that order, like symptoms and then lab stuff. Or do you want to actually?
For the first one, above 10%, it's okay to put symptoms and
Oh, okay.
laboratory abnormalities. For
Okay,
immune mediated, you don't need to just put that in that order, symptom first and the lab next.
okay. And then you want me to do this for all the other codes as well, right? Including
Yes.
thyroid. Okay.
Yes.
Oh, another thing is that these ones are written to the first decimal point, but for this one is rounded up to the whole number.
Oh, yeah,
Would you
so
like
it
me to
has-
round these? or
no, first decimal
where
is
it?
always the norm.
And so these should be changed too, or
Yeah, that should be changed too, unless it's exactly 38. Even if it is, like, it's, yeah,
oh,
first decimal.
okay, okay. But everything else looks good to you. These plot swimmers, this as supplemental.
Uh-huh.
Oh, this I'll have to check with Liam and the team.
Okay.
But yeah, other than that, it should be good.
Did you make that a table or is this a figure J file?
This is a table. Grace made a separate table.
Oh, okay, good.
Gotta switch this. Okay, any other comments for BAC?
I think this is, if you just change that, it's good to go. You don't, for, yeah, desmoid and this, you don't need my permission. You can just, go by the protocol, submit it with, Liam.
Okay. Thank you. And this is Volver.
What's left?
This is-
Bar and what are the ones?
Volver, non-epithelial ovarian tumor, non-epithelial ovarian cancer, and then small cell ovarian cancer, and this is clear cell gynecologic cancers.
Okay,
So we have 4 more.
okay, let's take a break. I gotta answer some texts and pages. And then we'll come back. Okay, I am back.
Here, I wanted to just ask you a quick question regarding like organizing the adverse effects. Should I just do symptoms and conditions? Because sometimes like I'm not sure where, because like hypothyroidism, adrenal insufficiency, like that itself is not symptom, right? It's the condition that can cause those symptoms.
Mhm.
Should I just categorize them as symptoms, conditions? Would you be okay with that?
Conditions. Yeah, that's fine.
Okay, and then laboratory abnormalities for like lymphocyte count this decrease.
Mm-hmm.
Okay. Okay, so this is Bulwer. Everything else should look the same. And then, yeah, I'll just clean up the first effect. Yeah, everything else is kind of the same. Then we only had squamous for the same.
Mm-hmm.
So yeah, this one should be ready as soon as I fix up the tables.
Yeah.
Finish the decimal points. Is non-epithelial. This one is JAMA short reports. For Volver, it was JITC short reports. This is JAMA.
Mm-hmm.
Yeah, everything is the same. Oh, this one I wanted to ask Liam, because I need the actual like data PDF file, and then I'll create a separate table instead of plugging in these screenshots. Same thing for the adverse effects. And then these resolutions are really bad. This one, it even might cut out the C part while I'm editing. And then this also has to be fixed. But yeah, other than that, this should be ready as soon as I do those minor edits.
Okay. So yeah, same thing. You can work with students
Yeah,
and acknowledge them. And at the same time, just tell the student why we're doing this and what this means, and so they can learn from this.
right.
Okay.
I'm just doing this in case I forget. Yeah, so same thing here. Yeah, again, need to ask me in for the actual files. And I'll update everything. Yeah, I'll update this as well.
Go back up to the toxicity. Is that yeah. So same thing, same rule apply.
Yeah, of course. Right. Right, that's that. This is a clear cell guide.
Yeah.
Let's see. It's Nature Communications. Yeah, everything is the same. This one I'll confirm with you.
Oh, yeah. So the other thing is, Liam, when you send the papers to Kurtrak and Patricia, always say this is formatted into what journal format, we're submitting to what journal, and also this was previously presented at what meeting, poster, oral session, or so that they know the history.
Okay.
Yeah, so for these ones, we have 3 subcohorts. And so we presented it this way: 46, 45, 42. The response outcomes. We'll do the same for these adverse effects. Plots. We have two different, one is for clear cell cervical cancer, one is RECIST based, one is iRECIST based. Randomized plot, survival curves, and then supplemental table. This one I'll confirm with Liam as well. I'm not sure why this is.
Yeah, check the path report with Liam. It has to
Then-
be clear cell, otherwise it's why is it?
Yeah.
Yeah.
So yeah, I think that's all. That's all for now. Yeah, those are the 6 colors. Yay.
Okay, so almost there. It's just a lot of formatting. But we need to get this out ASAP, like I said, because we're writing up a overall DART paper, meaning like we're writing up the whole DART study, regardless of the cohort. So it doesn't make sense to get this getting these published after the overall.
Hello?
Did he freeze?
Okay, so
Do you hear me? Did I get cut off?
yeah.
So I gave you list of the priority, like which cohort has to come out first.
Oh.
Remember?
Yeah, the list.
Yeah. So follow that list in terms of
Okay.
getting it out, because you can't do it simultaneously. But you can divide and conquer, let different students work on different ones, but give them deadline, but the ones that are high priority, get them out ASAP, because we have overall DART paper coming out very soon. So we want each cohort paper to come out sooner than that. So that's that.
I wanted to update you a bit about the Tempest collaborations for the
Okay.
Discover Mucinous project. The last time we heard back from them was June 26th, and they said they still need to have some more discussions between their medical data and legal teams to determine how, the agreement will proceed. So I didn't reply to them yet because I wanted to know if we are still going. We would still like to proceed with the, like the sending fellows route, or are we just going to wait for them to give us the data? Because I think initially the idea of sending fellows was because it was taking too long, but I think now they're running into a lot of issues. So just wanted to confirm with you if you still want to go with this route.
So I don't understand the question. So there's two, mucinous and the heterogeneity, right?
Right.
So which project is this?
This is the Discover Mucinous project.
Discover mucinous. And because they were taking too long, we said we'll go and learn. And they said wait. And I don't know where we are.
Yeah, I did all the background checks, and then they still said they'll need to do further discussions with their legal team. That was June 26th.
Okay, just follow up again, just say, we and Dr. Che want to, move this project forward, ASAP. Let us know if it's okay for us to go there and learn and extract the data together. If not, let us know when is the expected arrival time of the data for us. Just push them. I mean, I think it's probably not their priority. So I think we just have to push them, weekly basis, okay, to a point where they think like, wow, this is so bothersome. We need to get this done,
Okay. So since Dr. Xian will be gone, I think last time you said include Liam. So will it be Liam and me?
Yeah. Without Jian, you just both of them, you can go and learn. Pick a day and then just go and learn.
Okay.
Yeah.
And then TAP project, they said they were supposed to give us the data by the end of June, and they still haven't. So I will do the same thing.
Yeah,
after
same
their
thing. Just
ETA.
CC me on all that communication and, we're still waiting. Where is it? When can we start? Just,
And then lastly, for the last trial, Agenus, I'm not sure how to pronounce it, but Agenus, they still haven't gotten back to us. I uploaded everything on the portal, and it's still under review. But we will be meeting Akizo today at 1 p.m.
so
to
this
discuss
is
the last trial.
Asian. This is Asianus.
No, this is Akizo, the Chinese company.
Oh, the Chinese
It's
company.
like,
Can you pull
Was-
up the company's website?
yeah, the drug was AK-104, and then I will pull up the website.
Akeso Bio. Okay, Akeso Bio. And is this bi-specific or two drugs?
This is bispecific.
Okay. And are they phase 1? Can you pull up their pipeline?
Like for AK-104?
Yeah. Phase 2, 1B2.
Yeah.
What is the latest update? Let's look at that. Latest
This
update.
one.
ASCO, this is 2022. This is, oh, is this cadonilimab, okay. And pretty high response rate. Is this melanoma?
Cervical cancer.
Cervical cancer, okay. Okay, pretty high response rate. Overall survival not mature, so they didn't report it, okay. Yeah, that's pretty decent regardless of PD-L1, so. Okay, so maybe they could be a good partner, okay. So, the slide we made, just modify it for cadonilimab.
Yeah, I already did.
Yeah, so we'll just present that and see. And Xencor and Agenus, they're not responding to our email, both of them.
Well, agent has told us to just submit everything to the portal and just
Oh.
wait. And that's what we're doing. And Xencor has not gotten back to us.
Wow.
Has never gotten back to us, I think.
I mean, we had that teleconference about thyroid, right? Xencor. It was Xencor.
Yeah.
So it's surprising. So why don't you email them again, CC me again, that, how do we submit this proposal? Can we do a Zoom meeting? Please let us know how to proceed. Just the same emails. One last approach. Like, I don't know what to do with this. They don't have booth and yeah. So I think we had a couple emails with their company, right? So just resend that, CC me again. I think AZ is not interested. What are the other remaining companies? Can you show me that list?
Yeah, I'm looking at it right now. Here. Okay.
Share it.
Okay. Oh, actually, one question before we proceed. Do you want me to write AK-104 or the CADO? Blah blah blah.
It's okay.
It doesn't matter. Okay.
Doesn't matter.
Okay. Yeah, that's the list. Everyone else have declined. I think.
Yeah, they declined, right? Yeah.
Yeah. There's just these two.
Okay. I think we're doing our best. I don't know what else to do. Semi-pli map. Okay, I'll give you a homework for you. One more. So if you go to NCI CTEP, CTEP homepage, there are drugs. There are drugs applied by CTEP. So this is an agreement between CTEP and drug companies. So this is how I got nivo-ipi, the DART study. So the BMS supplies drug to CTEP, and then CTEP supplies a drug to the investigators in SWOG. Okay, so this is not a direct drug company to investigator collaboration. Okay, so see if there is any CTEP drugs that are PD-1 CTLA-4 combo or bispecific or two, like it could be nivo-ipi, it could be anything. See if there's any CTEP drugs. Then if so, then I can write a SWOG proposal to use that CTEP drug. Okay. So
So we have,
there's this MDX. What is this? Oh, this is just ipilimumab.
yeah,
Yeah.
but no, I don't see any combination drugs. We have Nivo, Pembro, Atizo, Durva. Yeah,
What
I'm
about
looking at this list of agents available.
under clinical and non-clinical studies? What about CTLA-4?
It was just ipilimumab.
Nothing else?
No.
Hmm. So Xencor, I don't know what the best way is. Let's keep thinking what the best way to get that concept approved. Hmm. Okay. Anything else?
That's all. Once I finish the papers, I'll let you know, and then I'm continuously working on the transplant paper. And I hope to get that done maybe next week, maybe next week.
Yeah. So, like I said, get all the DART papers out. Then let's work on
Of
process
course.
generating papers.
Okay.
All right.
And then I'll send you the Kiso PowerPoint file.
So from our students and Dr. Tegyu, I will get updates later because I have wait, we have 5 minutes. So Tegyu, at least you can give me some updates on what you've been doing.
Okay. Now I'm currently working on radiomics, and we're trying to get all the segmentations done by tomorrow. And there's an an ISD form from the Novocure company that you wanted me to write a clinical trial form, and I wrote a draft by myself, and I'll hand it over to Dr. Liam soon. I noticed that there is a meeting with Novocure on July 20th, and should we have to get it done everything by then?
July 20th. July 20th? I don't have.
I received an email from Novocure like this. Wait a second, please.
I don't have anything on my calendar.
Oh, a doctor named Susie Rice sent me this schedule.
What day?
Um,
July. July 20th is my clinic day. I cannot have any meetings.
maybe Dr. Liam has also received the message, the mail, or it says that you and Rebecca and Liam received this mail from Susie Rice.
So I have it for July 19th, 12 noon.
Oh, like I think my laptop is set up for Korean time, so maybe that's the reason. So yeah, it's not 19th.
Okay, yeah. We need to have proposal by then. So just work.
Okay.
Yeah.
Mhm.
And so again, those of you who are here, don't wait till Tuesday. You can always print out what you've done, show it to me anytime you run into me, okay?
Okay.
Okay.
Dr. Chit, can I just ask you a really quick question? We have an email from Janssen about one of your speaking engagements.
Lung cancer treatment education daughter. Okay, so just forward that email to me then. I will respond.
Okay.
Dr. Che, I've got one quick question. Oh, sorry, go ahead, Liam.
Yeah.
One quick questions. So, Garden got back to me after I sent the email and CC'd everybody. This is Leslie B. She just wants to meet and discuss kind of the current state of data feeds between Garden and Northwestern. Do you want to be present at that meeting?
And this is between them and our team?
Yeah.
Yeah.
Okay, so I'll reply to that email and I'll CC your Leslie for your availability.
No, so that my Leslie's gone. It's Susie.
Okay.
So
Susie. Okay.
Reyes, you can see that in my email signature.
Got it. Okay.
So have Susie schedule that meeting soon. Could be tomorrow or next week, Tuesday or Wednesday.
Okay.
But let's shoot for tomorrow. Have time tomorrow.
Okay, perfect.
And Liam, you can help. Like I have yeah, Susie can help. Yeah, Susie should be able to help. It's an urgent meeting. We need it preferably tomorrow or next week, Tuesday or Wednesday.
Okay, great. Will do.
All right, very good. So we'll wrap up here. Thank you, everyone. Great job.
Thank you.
Thank you.